Hi everyone, the gene that I was given was MYH7, which is also known as myosin heavy chain 7. I wasn't able to find any cool pictures that really relate to this gene, so my PowerPoint is going to be pretty simple. Again, I just have the MYH7 gene. The function of this gene is to produce a protein which is known as beta myosin heavy chain. It doesn't always happen that way where it produces that protein. This type of gene is a missense mutation. It is located mostly on chromosome 14. They found some bits and pieces on other chromosomes, but I don't think it was really that important to include that due to it's just bits and pieces. Also, it is found in cardiac muscles and type 1 skeletal muscle fibers. These fibers are known as slow twitch, and they are slow twitch because they are twitching at a slower rate than versus the type 2 skeletal muscle fibers, which go at a pretty fast rate. These type 1 skeletal muscle fibers rely on aerobic respiration. And then on my blast results, my E value was a 0, which was good because that means that it is an exact match. My identity percent was 99.95%, which is really good because you want that to be close to 100. My gap score was 0 out of 5,805, which gives you a 0%, meaning that this gap score is really good because you just want the percentage closer to 0 or that score to 0. For my enzyme and plasmid selection, I selected the vertebrate plasmid, which is PSFSV40. And on this plasmid and my gene plasmid vector, I was able to find the two enzymes known as STUI and BGLII. The STUI enzyme that I found cuts at the code AGGCCT, and my gene has 5 of those. On the BGLII, it cuts at the code AGATCT, which I was able to find 2 of those cuts on mine. For the selectable markers, I have CAN-R early genes and late genes. CAN-R is kanamycin resistance. Early genes are just proteins that are in viral replication, and late genes are viral capsid proteins. So the organism that I selected is known as B. taurus, which is just Bos taurus, and it's commonly known as a domesticated cattle. I chose this because I read some things where they did some research on it, but I just kind of gave my input on those within the next couple of slides. For my transformation method, I chose the lipid-based reagent for vertebrates, and I have this little graph that kind of makes it a little bit more simpler to understand, in my opinion. So in the first kind of step, what it does is a phospholipid has a hydrophilic head and a hydrophobic tail, which means that this hydrophilic head is going to be negatively charged, and the hydrophobic tail will have no charge. And so since there will be a transfection reagent entered, that reagent will be positive, and so obviously a negative and a positive attract each other. This nucleic acid and the transfection reagent is going to form just a complex, this nucleic acid lipid complex will enter the cell, and then the sequences will be released. After they are released, they will go ahead and head into the nucleus, causing this lipid-based reagent for the vertebrates to be successful. So the potential impacts on the organism that I chose, which was the domesticated cattle or B. taurus, and the impacts on the environment would mostly be negative in my opinion. I know we're supposed to have some positive impacts, but just with this certain mutation, there's not really anything great looking. There's not really like a great outcome for it. Therefore, I will discuss some positive impacts that I think would be possibilities. I'm not sure if these things are being done yet. With technology now, there is a great possibility, but if everything that I read, I wasn't able to find anything that was that great. But first, we're going to focus on these negative impacts. So if we were to take the MYH7 gene mutation and enter it into a cattle, the meat quality would be pretty bad. It wouldn't be as tender, and the flavor would just be completely off. It wouldn't taste right. I do know that in humans, the MYH7 causes a lot of heart problems and muscle problems. These heart problems are stuff like cardiac arrest, heart failure, and the muscle problems are known as myopathy, which is a disorder of the skeletal muscles. And there's a lot of causes. This can be genetic, which could be the reason MYH7 is linked to this, because it's, in a lot of the research that I have been doing, it is mostly genetic. But if these problems are able to occur in humans, I would assume that cows are able to get it as well. I'm sure that they can have heart problems. I'm sure they can have muscle problems, just as us humans do. So if they're having all these problems and our meat is turning out bad, or say they are having heart problems and end up passing away, we would have a decline in our food chain, which it wouldn't be that dramatic, but it would have a pretty hard impact with it. With beef being one of our top meat productions that we have. As far as the positive impacts, I think that we could have some great possibilities in medicine. If this gene were to not be mutated and act properly, we would be able to save cancer patients, I feel like. If not, we would be able to at least help find some type of medicine to alleviate some of the heart and muscle problems. As far as the cattle, I'm not really sure what a positive impact would be. If this was like a perfect world and everything were to happen so great, I think that we could use this as medicine on the cattle as well to keep them alive. And if we're keeping them healthy and they're able to breed, then there would be more food possibilities, which would increase the food chain. Well, not really increase the food chain, but there wouldn't be, we wouldn't have to worry about resources for the beef production. But again, I think that this is something that could be done in the future due to the technology that we have today. But as for now, there's not really anything that I'm seeing that is positive on it. Here's my works cited page, and I hope you guys enjoyed my presentation.